This is the last of the biomolecules that we have to worry about. We're breaking it down at least. And I have put the one with the blanks. Oh, by the way, send me an email later on today and I will put the unadulterated, that sounds awesome, unadulterated PowerPoint for Chapter 17, Part 2 on my fire. All right. But, yeah, so the one with the blanks for Part 1 for Chapter 18 is already available on my fire. So are the suggested problems. So I just had this in there because I came from the Parks and Recreation. It's an old show now, but that is awesome. They had me meet Tornado. So we're talking about proteins, breaking down proteins, getting energy from your proteins. Which, by the way, one thing that keeps proteins a little different, we have a way to store fats, right? We have, those are lipids, we have ways to store carbohydrates and sugar, particular glycogen. But we don't with protein. I mean, yes, you can say muscle, but you don't want to build muscle just to break it down for the energy. That's not the whole purpose. So we don't store protein per se for energy storage. Okay. So it's one that is only in the shorter term. So this is called oxidative degradation. I just abbreviate it. If it says AA from here on out, it's not Alcoholics Anonymous, it's amino acids. Okay. That way you don't have to go and tell everybody we're talking about AA and biochemistry. So, what are the main parts of the amino acid that can be degraded? Oh, which, by the way, I just realized that one of the slides didn't come through. Some little factoids for you. Carnivores can derive up to 90% of their energy needs from amino oxidation if it's right after a meal. You just ate, that double patty grilled chicken breast sandwich. You can use up 90% of that for energy. Little other thing. So there are only 3 circumstances you get your protein-rich diets, but once again, unlike fats and sugars, you can't store protein. You can have amino acid oxidation degradation. It's called oxidative degradation, I'm sorry. Oxidative degradation just from normal protein turnover. Right? You have to have it, there's a, just like it says in Ecclesiastes, there's a time for everything. So a time to be born, time to die. Yeah?
Sorry.
You still think about the meat tornado?
Yeah. So if you don't eat carbs with your protein, then you're just going to be using those proteins you eat for energy sources. Is that what you're saying?
I mean, you're going to have to get energy.
Right.
And so if you're only having a primarily protein-rich diet, well, even just for an immediate meal or whatever, then the first place that you can get your energy may be from stored. Right? You don't store enough glycogen to do it for very long, and most of it's going to go for your brain. Then you're going to get fats. That's one of the reasons why the Atkins diet does work, especially in the short term. It can work. You can lose fats, storage, pretty fast, especially for men, sometimes more so than women, because the fact is, if you're only eating protein, there's no way for you to store protein for long term. And so you're going to get it from energy, and then you're going to start having to use up your fat. And then at some point in time, which we'll see that, and that's point number 3, it can go out of whack. It can be bad. That's where, if you had to choose between me on my soapbox, if you had to choose between the Atkins diet versus the South Beach diet, which is where you do protein, high protein, but you also supplement that with certain types of carbohydrates, then that usually makes better sense for long term. But yeah, so the answer is you're going to have to use it for energy. At some point in time, if you have very, very low percentage body fat, then you're only on a protein-rich diet, then it's going to have you're just not going to come protein. Okay. Because in the third thing, so you get there are 3 times you get what's oxidative amino acid oxidative degradation, and that occurs 3 ways. 1's high protein-rich diet. 2 is just from normal protein turnover. It's not the same from Ecclesiastes. Proteins have got to, when proteins start to go bad, they've got to be degraded. You can break them up from amino acids. You can also just, that's just normal protein turnover. And the third way is through starvation or uncontrolled diabetes mellitus. It's called ketoacidosis. Ketosis. And if it gets out of whack, then that can be really bad. And we talk about that later. I think it's later this chapter. If not, it'll be in the other chapter, but we'll talk about ketosis and ketoacidosis and how ketone bodies can be bad in that sense. Okay. All right, so what part of the amino acid can we use for energy? Okay, and so, just generically speaking, and I would expect you to know what an amino acid looks like. I'm going to do it as a carboxylate. Okay, this is the generic amino acid. If we go part by part, what parts of it can actually be used to get energy?
The R group, depending on what R group is?
Depending on what the R group is. Possibly. I mean, if we break it down, can you get energy from this hydrogen? No. Okay, can you get energy from a carboxylate? You get very little, because carbon dioxide, CO2, means that carbon has given up all of its energy. And we've got to get rid of this. This is the whole purpose. This is bad. Bad. Okay, so the nitrogen in and of itself, if you try to get rid of it, as ammonia, you would die, right? If you got too much ammonia. We're not fish. We can't just have it seep out of us. Just because ammonia-like animals, okay? And so it's really primarily from the carbon backbone, depending upon the R group and that. So you're really limited in that instance. All right. And so what we're going to leave off on for today is you first have got to lose the amino group, because you want that alpha keto acid, because it's going to be the alpha keto acid backbone that's going to be the important part. Then from that carbon skeleton, you only have three possibilities. One is you can oxidize it all the way down to carbon dioxide and water. Like I said, carbon dioxide means that carbon's completely spent. You used it, I should say use and abuse, that's bad. That you used it, you're going to get rid of it. Kick it to the curb, so to speak. Sounds like a really bad Springer episode. Then you're going to try to break everything down into 3 and 4 carbon units, so that way you can convert it ultimately to glucose, which what metabolic pathway will take those 3 to 4 carbon units and put it to make glucose. What makes glucose? Gluconeogenesis. Glycolysis will break it down, but gluconeogenesis can make it from the liver to go ultimately to the brain, for the brain to take it for glycolysis. Or you can break part of them down and put them into the citric acid cycle. We're going to try to see, and that's the kind of thing that we're going to see over and over again. And then we will talk about, there's got to be a way to get rid of the amino group. For us, we use the urea cycle. But not all animals use the urea cycle. It's the reason why birds don't pee, which I didn't realize that not everybody knew that, but I had one student that swore up and down that they saw birds peeing. No. They're eureka-tail-like animals, so they do not. They don't have urea. So, yes, you won't see that bird highway to do that. All right, another thing for HQ. You guys are going to be on fire today, so let's just pick up. So this is the overview, like the big, broad scheme of things. That's why sometimes, like whenever I took biology many, many years ago, they even referred to it as the Krebs bicycle. Because the Krebs cycle, it's a citric acid cycle, but it is linked to the urea cycle by this shunt, which they show as a third cycle. So it would be a tricycle, but that's neither here nor there. But yeah, it's called a shunt that joins the two. And so we'll talk about what that means later in this chapter. But we can you have the proteins are broken down to the amino acids. We've got to get rid of the nitrogen. And it depends. You can either use it, there is some biosynthetic uses for it, but ultimately you're going to have to try to get rid of it. And for us, we get rid of it in urea. But then the carbon skeletons, we make the alpha keto acids and ultimately get that for the energetic intermediates. Okay. This figure right here is very important to understand. Okay. I thought I had one that had blanks and stuff on it, but there were so many blanks to it that you couldn't understand what was actually supposed to be there. So I must have made it for an exam at some point in time or quiz. But then I couldn't remember what it was supposed to But it was missing stuff, so I couldn't put it up there and give it to you guys.
All right. But this is very important. So we have, it doesn't really matter for me where we start. Actually, I'm going to start down here. We can have, alanine, and we'll talk about the role that alanine plays later, but alanine can come from the muscles. And once the alanine comes in, once again, we can actually use an aminotransferase here, in particular be the alanine aminotransferase, in order to make pyruvate, which pyruvate can be used, energetically as well. And also, it's going to make glutamate, okay, the way that they have this one drawn. Because the glutamate and alpha-ketoglutarate is the pair that's utilized with all of the aminotransferases. That's why we don't have a glutamate aminotransferase, because glutamate is used for all the aminotransferases as the pair between these two. Okay. Now, if you had just any of the other, including alanine, but any of the ingested proteins, any of the other amino acids, we have the amino acid here. And once again, it can be formed to make the alpha-keto acid, and it would give its nitrogen to glutamate, and we just repeat this process over and over again. Ultimately, you're going to get rid of the nitrogen. The nitrogen is going to be released either in the form of ammonium, urea, or uric acid, depending upon the species. And then we also get glutamine. And glutamine is another way to traffic nitrogens from tissue to tissue. And the glutamine will then ultimately get rid of its nitrogen. We'll talk about the process with which that happens to make glutamate. And so we're going to see how glutamate is involved in multiple pathways here. Okay. We're going to focus on this part right here. There's the ammonium, the urea, and the uric acid. Okay. So the three different broad classes. In the waste products, there are those that are called ammonotelic, there's ureotelic, and then there's the ureotelic. Okay, so this is the reason why, and usually people can, for whatever reason, remember my statement about how birds don't pee, because birds do not have urea. Okay, so I'm actually going to start with the fishes. Sounds like, never mind, I won't go there. Okay, I had a song in mind. All right, so most of the aquatic vertebrates and fish, they can actually just release their nitrogen waste product as ammonia or ammonium, okay? And so that's quick and easy to get rid of. For us and sharks, and don't ask me why sharks are different, but for us and sharks, we're going to get rid of it in the form of urea, okay? And then birds and reptiles, typically, they're going to get rid of it in the form of uric acid. That's why they're called ureotelic animals. And so if you notice, all of the carbons are highly oxidized. Why is that important? And the same thing can be true with urea. It's highly oxidized, that carbon. What does that indicate? We used it for all it's got. So do the birds. So, yes, we got all of the electronic energy from it that we possibly could, and that way we're not wasting. Okay. All right. This floor is making noises over here now. All right, so now let's start to address what happens to the amino group. We have two really important classes of enzymes here. Okay. That you need to talk about. One will be the enzymes that degrade proteins. So, number one, what class of enzymes degrade proteins? Those are proteases. Proteases. Okay. And then secondly, we have the amino acids themselves that are going to have to lose their nitrogen group. And what class of enzymes for all of them except for glutamate, what class of enzymes typically are used to remove the nitrogen groups? Those are the aminotransferases. Or transaminases for the old names, but okay. So we're going to focus first on the aminotransferases. All right. I forgot who I was talking with. Oh, Nathaniel. We talked about, equilibrium constants, and I couldn't think of anything off the top of my head that had equilibrium constant of exactly 1.
Well, aminotransferases are really, really close. They're really close. So actually, let's start with that, because you already know what the aminotransferase says. If it has an equilibrium constant of about 1, what would the delta G be, approximately? Because what's the equation that relates delta G and K?
KQ equals delta G minus RL, something.
No. That's delta G is equal to delta G prime plus RT. That was what the thing that?
Yeah.
RT ln of Q. There isn't a Q, except for that Q. There is no Q in this equation I'm thinking of. What relates? Really, you just need to know what function it is. You don't need to know about any constants. That doesn't really matter. What's the function between K and delta G? K is equal to 1, delta G would be equal to, or approximately 1, delta G would be approximately equal to 0. Okay, because remember, it has to do with the natural log of K. Okay, and so, what does that indicate? If delta G is about 0, or KQ is about 1,
Equilibrium.
you're right about it, always at equilibrium. Okay, so that's why it's easy for it to go both directions. All right.
So it does remove the amino groups from all the amino acids. And I mean, technically it does remove it from glutamate as well. It's just that this isn't the way to ultimately get rid of the glutamate's nitrogen, since it would just be handing it off to another amino acid. So it is, like I said, it is reversible. And you're always going to have an alpha keto acid, right? This is the ping pong mechanism, if you think back to last semester. Okay, so the amino group is transferred to alpha ketoglutarate to form what amino acid? So let's say if we go from alanine, alanine is going to make pyruvate by taking the nitrogen from alanine. It gives the nitrogen to alpha ketoglutarate, and you made what amino acid? What? Glutamate. Okay. All right, and so they all have the exact same reaction mechanism. They all use the same prosthetic group. It's called pyridoxal phosphate, or PLP. You do need to know, I didn't double check the most recent version to make sure it's figure 18.6, but the figure's pretty obvious. You do need to know this mechanism. For those who are in enzymology, we've actually already covered it in even greater detail than it's covered on this one.
And this is what PLP looks like. Pyridoxal phosphate actually has 2 different forms. There's the PLP, and then there's pyridoxamine phosphate.
It's this aldehyde that's really important here. All the rest of it is not as important. This aldehyde here easily forms imines, which are also commonly called Schiff bases. Okay, and this is an electron sink, because the electrons can flow down this direction towards this nitrogen. They can flow back towards whatever it is that we want to make or break.
PLP is covalently attached to the enzyme. Once again, it is reversible. And remember, that's called an imine. Imine means it's a carbon with a double bond to a nitrogen. Looks just like an acyl group or carbonyl group, except for the fact that it's a nitrogen. So it's a lysine that's on the enzyme that goes, and you can review it from organic chemistry how the condensation reaction works. But it goes and it frees up. They're showing this one step. In reality, you have to form a tissue, intermediate, and all that kind of stuff. How it frees up a water.
Okay. They do, and we've mentioned this before, they call this a Schiff base a lot. You see these over and over and over again in a lot of mechanisms.
This one actually forms another intermediate. Not only does it form a Schiff base, but it also forms what's called a quininoid. And I'll show you the spelling for that in just a moment.
Okay.
I don't expect you to memorize the structure of PLP. but if I gave it to you, I would expect you to know how to use it.
I had to memorize all these structures. That was a pain. All right, so PLP. There are 3 types of reactions. Aminotransferases actually do more than just aminotransferasing. Transfer of amino groups. They're also involved in racemization, which, remember that just means that you can switch between the different enantiomers, D and L. And they can do decarboxylation reactions. And if it utilizes PLP, you can do decarboxylation as well. But for all of them, there's that shift base and that quinonoid intermediate. This is the figure that I'm going to show you, that I want you to know that the mechanism, technically there's 3. And I will break it down on each of the following slides. So don't feel overwhelmed, immediately at least, don't feel overwhelmed. Okay. Let me make it a little smaller. Like I said, this is broken down on each of the following slides. And I'll walk you through it, but I strongly suggest that you know how to do these and that you take some time to work through the mechanism yourself over and over again if need be. The top one in the box, the common aminotransferase reaction. And one thing I wanted to point out that I don't care for about this mechanism, the way they have it shown, is, remember, this is called a ping pong mechanism or double displacement. It's only showing you the ping, not the pong. The pong essentially is the reverse of it. And so that's why they're showing it being reversible. But that's why, if you notice, you don't get your enzyme back. See, this is where your enzyme is, but it's still stuck here because you got to go backwards. So that's just one little thing to keep in mind. Where in the other 2 forms, you do get the enzyme back. But this is the aminotransferase. This direction here is racemization, and then going down like this is the decarboxylation reaction, which I didn't check the most recent edition, but in the previous editions there was a typo in this one. Okay. That I made note of. All right. So here we go. This is the aminotransferase reaction mechanism, and it's very important. And it utilizes a couple of important points that I wanted to show. It likes to, the aldamine, you kind of get an idea. You know, we talked about aldols, there's an aldamine and imines and all that kind of stuff. It's just where we have an aldehyde and imine. Okay. So, in the beginning, we have the amino acid that we're trying to get rid of its nitrogen to make the alpha keto acid form. We've already shown how the enzyme has used a lysine to add on this prosthetic group, the PLP, and there's the imine. One thing that is not shown here, and it's just assuming that you know, is the fact that this nitrogen can supplant this nitrogen. And so if you notice, the enzyme disappears, and the reason why is because this nitrogen comes in, smacks that carbon a couple times, and it frees up the enzyme. But this is still withheld inside the active site of the enzyme. It's not going to float around. And so that's why we now have the imine forming between what had been the amino acid and the PLP. Then we have that general base. Think back to last semester. Remember, general bases, according to the definition for Bronsted-Lowry, means that they are proton acceptors, whereas general acid would be a proton donor. And so all it does is it has to have free electrons. Obviously, it's a nucleophile. Comes, smacks that hydrogen, and it only takes off that proton part. You know, boom, sound effects optional. These electrons have to go somewhere, and so they're going to flow down that sink of the shift base. So that's what this is showing. It's going to go to form a new double bond between the carbon and this nitrogen. That double bond breaks to form a new double bond between the 2 carbons, and then this double bond will form a double bond here. That double bond will go to make the free electrons. That's why this is called a quinonoid. It is related to coenzyme Q. It is not CoQ, but that Q stands for quino whatever, you know. And we're going to see that in the chapter on electron transport chain, that these quinonoid intermediates
in like the CoQ intermediates, you see this kind of ring formation to where the electrons dance around the ring, ultimately. Okay, so that's why it's. Just want to point that out. And when we get to coenzyme Q, I wouldn't expect you to memorize. So coenzyme Q has a quinol, quinone, and semiquinone for its quininoate intermediates. Okay. All right. So now the electrons are all down here. Not all electrons, but the electrons have flown down the sink, so to speak. And then what we do is we get the reverse, which you could go all the way back, but if so, you didn't get rid of that nitrogen. So what happens is we're going to reform that double bond. That double bond breaks to reform over here, and then this double bond that was here takes a hydrogen from a general acid. It could be water. It could be an amino acid group there. It could be the BH, the H off of that. You know, it's going to depend upon what's in the active site there. And so now it looks like this. So this is just a common rearrangement. Okay. Now, one thing that they're assuming that you can remember from organic chemistry too, and so on your own you may want to review, this oxygen is going to be a nucleophile that can come in and smack that carbon, essentially it does it twice, in order to free the amine, and you have your alpha keto acid. So it's water that comes in ultimately to replace and to knock off the alpha keto acid. So in effect, we've gotten rid of that nitrogen. But that nitrogen's now stuck to the pyridoxamine, the PLP. Well, it had been PLP. Technically, it's not pyridoxal phosphate anymore. Okay, and so then what you do is you're going to go and do a reverse of that in order. The next, so if this was now maybe alpha ketoglutarate comes in, alpha ketoglutarate goes backwards to make glutamate, essentially. And so that's one way to go from an alanine to make pyruvate, then alpha ketoglutarate comes in to make glutamate, or vice versa. That's just one example. So again, please go over that mechanism, well, all three of these mechanisms. Because there's a couple of things that we may see over and over again. We've actually seen shift bases before, but we see these shift bases. See, there's one there. We have it here. And, well, those are the important ones. Oh, and technically here. Okay. And then we have this quininoid, where you have essentially the electrons can flow down towards that nitrogen and then flow back. Reform the double bonds, reform them again. Or the reversal of them, I should say. And if I were you, at least one time, you may want to work it out to make sure you can go from exam backwards. Or maybe you want to like practice like showing the swap between like an alanine to make it glutamate or something like that. But remember, glutamate, alpha glutamate is always going to be one of the pairs. Okay. Any questions over this before we show the racemization? Racemization is pretty easy. Questions?
So you're saying an alanine can come in and then make the alpha keto group?
So if this was alanine to begin with,
Then it would make pyruvate.
it's going to make pyruvate, and the pyruvate leaves.
Yeah.
That's the ping. Never got to pong it. And so then alpha ketoglutarate would come in and go backward to make glutamate.
Okay.
Whereas if we started off with oxalic acid, let's say now we have glutamate. Glutamate would come in, would make alpha ketoglutarate. Then maybe oxaloacetate comes in and would make aspartate. Okay. Those are the two sets that you already know their name for.
Would we have to show the hydrolysis? We have electron.
Typically no. Okay. As long as you understand like what's going on there. Okay. Now for racemization, that's what happens. Let's say that we have an L amino acid, we want to make it a D, or you have a D amino acid, you want to make it an L. The first step here is the same. You have to form this imine. Okay. So for the decarboxylation reaction, you actually start right here, but it's going to look a little different from this point. So remember, you have our amino acid, and the whole purpose is to get rid of this carboxylate. That's why they're showing it that way. The first step is the same, but then That's why it's very, very similar to what we've just talked about. And you get the reverse, just like what we saw before, to where we made the hydrogen, except the big difference is the hydrogen, instead of it being added to this carbon, the double bond forms up there, this one breaks, and so the hydrogen's added to what had been the original carbon of the amino acids, the carbon of the PLP. So you just move one carbon down,
And then the enzyme, lysine, comes in and just replaces this lysine. This nitrogen, I said lysine. The nitrogen from the lysine here comes in, smacks that carbon, and will free off. And so essentially you went from an L to a D or a D to an L. Okay, because it's the same amino acid, you just changed the stereochemistry because that hydrogen got added on to the other side. Okay. So there is no water there, because it's the nucleophile that came in to knock it all off, was the enzyme. Any questions on this one? Like I said, if you know the aminotransferase, this one's not too difficult. The decarboxylation one is completely different, weird. Okay, let's see. I'm going to actually go back a couple slides first to show you where we started. Now let's go forward. Okay. All right, so I couldn't remember if I left that on there or not. All right, so what happens here? Oh, and then it's missing a hydrogen on the carbon. Like, see, the carbon, it lost the hydrogen. That hydrogen's still there. I don't know why why it's gone. Okay, that's an old typo. Okay, but no, what happens now is when we form this initial shift base, then instead of the way the electrons flow here is we get collapse of the carboxylate. So these electrons move down, boom, sound effects optional. So that's where you're going to make carbon dioxide. Then this bond has to break, and it's going to form the double bond now between the carbon from the original amino acid and the nitrogen. This double bond goes down here, then that double bond, ultimately flows down to make the lone pair electrons and the quinonoid intermediate. Once again, there should be a hydrogen there. It's just not listed. Then you get reverse. Okay, so you reform the original double bonds of the ring, and this double bond will reform that imine, and this will swing out to pick up a hydrogen from a general acid. So therefore, we have this nitrogen. And then the final step is that the enzyme comes in with its lysine, and it smacks that carbon and will ultimately free off the amine, and it has the paradoxal group on it already. So now this is one way, since you decarboxylated it, you made an amine. And we're going to see this kind of reaction later on, especially when we start talking about neurotransmitters with like histamines and things like that. We're going to see these decarboxylases. Mm-hmm.
How does the enzyme regulate between decarboxylating or transaminating? Like what's to stop when it tries to transaminate, the decarboxylation to occur?
Well, just like with dystrophin that I've heard someone recently speak about, you can have different isoforms and different types of it, so then they have different functionalities. Same thing can happen with enzymes as well. Or we've also seen where an enzyme can have multiple functionalities just based off of either compartmentalization, the ratios, like almost like what's more need, or they could be competing, but it's just like which one breathes out first. But typically the decarboxylases that do this will be separated from the aminotransferases. Okay. Any other questions on this? All right. But we have a problem with glutamate. Let's say we no longer need it. We're really trying to get rid of those nitrogens. Ultimately, we've just been passing them around. It's like passing the buck. Glutamate's going to get it. Okay? But we're going to have to eventually, how do we get rid of glutamate's nitrogen? Okay, you can't use an aminotransferase. So think about, why can't we use just a normal transaminase, normal aminotransferase? Why can't you use that with glutamate?
You're going to generate glutamate.
You just generate the other amino acid, like alanine or serine or whatever. And so you have to have some other way for this to happen. And so it's got a great term. I like this term. It's called oxidative deamination. If you say it really fast, it kind of sounds like something that would be a good point in a sermon. Damnation, that kind of thing. But yeah, it's oxidative deamination. All right. And we have to compartmentalize. This is where, once again, it depends. Ultimately, you've got to get to the liver. The liver is what's going to help undergo
the oxidative deamination here. And in particular, it's going to be the mitochondria. Okay. The hepatocyte mitochondria. So just like I've talked, and hopefully you're keeping up. Every time you get a new enzyme class or family, you add it to your list. Remember, I also suggested maybe having a big piece of paper, whatever, that's got like parts of a cell to it, and you can kind of compartmentalize like where glycolysis is occurring, where gluconeogenesis, that kind of stuff. Well, this is oxidative deamination is happening in, or at least this part of oxidative deamination is happening within the hepatocytes mitochondria. Because it's urea, the urea cycle is kind of like gluconeogenesis with respect to how it occurs, how it's in multi-compartmental. Hopefully you understand it. I completely made that word up, I think. All right.
This is the reaction. Let me make it. I'll give you a chance to write it down.
We still are going to make alpha-ketoglutarate. Okay. Because if you deaminate glutamate, that is the product. It's just going to have to be a different enzyme. It's not an aminotransferase. So I'll give you a moment to catch up. A little side fact, factoid thing for HQ. This is the only known enzyme, at least as of this time, that accepts either NAD plus or NADP plus. Usually it's one or the other, or one isoform will take one and one isoform will take another. This one, just throw the NAD part. It doesn't care whether it's been phosphorylated or not. So.
Which, and you can even think of it this way, it's another way to make more energy too.
Because that NADH, the NADH, could go on to ultimately make ATP if need be.
Okay, what class or classes of enzymes can do this?
It's a dehydrogenase. So if you're going to name this dehydrogenase, what could you name it?
Glutamate dehydrogenase. It's pretty simple. This one's not too bad. Okay.
It's activated by ADP. It's inhibited by GTP.
Right.
So glutamate dehydrogenase is how you get rid of that nitrogen. We'll talk about the role that the ammonium here plays. We still, if you're a fish, you can just let it go kind of thing. But we can't. So far I haven't taught a fish. So, and I wouldn't remember, right? I've seen Finding Dory.
Okay. So, probably too often.
Between my wife and my son, I've seen a lot of Disney movies way too often. Right now we're on a Coco binge. So Coco helps him fall asleep, so we play Coco a lot. So.
All right. What? What?
They won like best song.
Yeah, Coco won best.
Yeah, Remember Me? Yeah. You didn't like Remember Me?
I just can't believe it beat
This is me with that.
Yeah.
It'd be what?
This is me, the Greatest Showman.
Oh, I haven't seen the Greatest Show.
You need to.
Oh, I'd like to. We've talked about doing some.
You should make a field trip.
And no, we can't do that instead of the biochemistry final.
Okay.
I'm going to give you a moment to look through this, because now we're going to have to start talking about how do you get the nitrogens or the amino groups, transport those through the bloodstream.
Okay. Because ultimately, we got to get it to the liver, but not all nitrogen starts there. We have nitrogen in other places. So how do you? Because you've got to get rid of it. And we'll talk about once again. I can't stress enough. What happens if you don't get rid of the nitrogen? Bad things happen. So first off, I'm actually going to start with the second point. I probably ought to do that differently. There's intracellularly versus intercellularly. If it's within the same cell, you can use glutamate. But we can't use glutamate to get from one cell to another cell. Externally. Not externally. Intercellularly. Because I don't want you to think that nitrogens are popping out of your skin and dead and things.
All right. Whoops, sorry. But ultimately, it's going to depend upon the tissues. We have skeletal muscles versus all the other tissues. Free ammonia produced in the non-skeletal muscles use glutamine. Okay, don't get glutamate and glutamine mixed up. Glutamine is what they will use. So if your brain, for example, needs to get rid of nitrogen, it's going to send glutamine to do it. Whereas skeletal muscles typically will send alanine. Why does that make sense? Why send alanine if you're a muscle? What's going on in those muscles? Skeletal muscles. Bilderberg. What do you typically have more of? Lactic acid, which lactic acid can then make. Pyruvate. Pyruvate can make. Start connecting the dots. What? Alanine. Right. And so that's why it can make sense. We're going to actually start now with glutamine, though. Glutamine is non-toxic. If you think about it, that makes sense. You know, glutamate does have an extra carboxylic acid. So, but glutamine is non-toxic. Glutamine is much, much higher concentration in your blood than all the other amino acids, which makes sense since it's the way that the other tissues are trying to get rid of their nitrogen. Okay, and by the way, glutamine, we're going to see a lot. We see glutamine a lot. All right, so give you a moment to look at this. You start with glutamate. So let's say it's your brain. This is your brain. That's well before from your time. Okay, but or whatever, a non-skeletal muscle tissue that's trying to get rid of its nitrogen. It has glutamate. Okay, it added it to make glutamate. So it added it to alpha ketoglutarate. It made it glutamate. You now got to get it out. Okay. Okay, so how do we do that? I'm sorry. This is going to drive me nuts. It's crooked. The first step, technically it's two steps, but it's the same enzyme. Okay, that's really important to point out. That's why I wanted to confuse you. The first step is that it phosphorylates it. If you notice, they call it gamma glutamyl phosphate just because in theory, glutamate does have two carboxylic acids that can be phosphorylated, and it's the one that's the gamma position. So it takes an ATP, it phosphorylates it, and then it'll take whatever nitrogen it wants to get rid of, and it's going to stick it on there, okay, to make glutamine. So what class or classes of enzymes can do this? Oh, kinase. What else could it be? What? It's a synthetase, right? It uses ATP, and we're building something. What are we building? What? Glutamine. So we put those together. It is glutamine synthetase. Remember, synthetases are just enzymes that are building, synthesizing something, but it does require an energetic intermediate at the expense of an ATP or GTP, I guess, in theory. Now the glutamine can go. It goes. This goes on. Okay. It leaves. It gets to the liver. This enzyme is very specific. It's going to be in your hepatocyte mitochondria. And it's going to remove that nitrogen. And yes, Nick, it knows to remove that nitrogen, not this nitrogen. Okay. It's very, very specific. And then for us, that nitrogen is ultimately entering the urea cycle. Okay. If it's fish, just let it go. What enzyme is this one? Does anyone know? It's got a special name. You know how we have like enolase and allolase and fumarase and those. So what can you name this one? Really close. Instead of glutamase, it's glutaminase, right? Because it's glutaminase. It's the enzyme that breaks off. It's glutaminase. All right. Any questions on glutamine synthetase, glutaminase, anything like that? Then we have here the alanine, which we've actually already discussed this. It's called the glucose-alanine cycle. It happens in concert with the Cori cycle that we already talked about with lactate, and like I said, we've already done. They can use glutamine, but they typically, if they're in use, they're going to be using alanine. And the reason why, just from what we've been talking about, for anaerobic conditions and everything. So I'm not going to go into greater details since we've already discussed that one. All right, so alanine, it is non-toxic as well. It should go without saying. You're going to have to, you can circumvent the citric acid cycle, and you need to ultimately replenish the muscle's glucose. So that's one of the reasons why it's going to be used in conjunction with the Cori cycle with gluconeogenesis, okay? Oh, right. And so on your own, you may want to review the Cori cycle that we talked about. Oh, Lord, it's been a while. Okay, a few chapters back, I remember. And then finally, reason why, at least for today's, reason why